Okay, just before we get going, which we will in a second, I know there's a couple of more. What a crowd. Oh, no, it's like a reunion here. Um, it's only because we have a famous speaker. Okay, I'm not doing those intro stuff. All right, so just to remind everybody on Tuesday, next Tuesday, there's a potluck to welcome all the new summer students. So as many as we have here, we will have twice as many people at these seminar series coming this summer. So we'll have to buy a lot of pizza. Anyway, it's a potluck though, Tuesday at noon up in the pod. You're all welcome. Please bring something, anything, come prepared. And do I have any other announcements to make? Caitlin's running solo today. All right, well, I guess not. So I'm going to hand it over to Chris. Sure, I'll leave the mic on, Chris.
So it's great to see such a nice turnout, and it's a really a reflection on the esteem that Tony now holds in the community. He is a storied and has stories about him. He actually almost had an alternate career as an NHL hockey player. And you can see from his nose that he decided to go to science instead, but he was playing AA hockey and then went to the States for the college hockey. And he did his BSc in honors at, I've forgotten somewhere. Where? A college town. And then he went to Sunnybrook on Long Island to do his PhD. And I remember when he came to my lab in 2011 until 2017, one day he said to me towards the end of his postdoc, Chris, it's so nice, being in your lab. Of course, right? Everyone loves being in my lab, right? But he said, no, it's so not. But that wasn't the reason. He said, it's so nice being in your lab. I don't have to lock up the freezers at night. And I thought he meant like the -80. There's a little key thing on the -70s, -80s. And I thought he meant that. And he said, no. We had to, at Sunnybrook, we had to lock up our freezer and our fridges every night before going home. Why? Because the other lab members from other labs would come in and alter your samples around to screw up your results. So you would fail, and therefore, by you, a zero-sum game, they're trying to play that game, that you look bad, therefore you look good. Sick. And that's not even the big East Coast and West Coast major universities. Don't go there. I'm not a great fan. But he also reminded me of when I was introducing him down at ISB, Institute of Systems Biology in Seattle, for a conference. And I said, I want to introduce to you, not to 70 people. I think this is one of his first big talks amongst a larger audience, to Tony Dufour. And I thought it was really funny, but only because his name's Dufour. Right, F-O-U-R, right, yeah. And so also then, like now, no one laughed at it. So one day I'll learn, right? But actually, I have no shame, so I probably won't learn. But also, I learned online. I mean, there's this wonderful thing called the web. He was in Canada's top 40 under 40 in 2020. So that means he's like 23, I guess, now, 24, 43 or something now. But so that was good. He's got that kind of fame. Did you get a lot of paparazzi coming around and a lot of people stalking you online, Tony, after that? So it was for science, it wasn't for being your looks or your glamorous lifestyle.
It was just science, yeah.
Yeah. And 2 years ago, he won from the Canadian Mass Spectrometry Society, the Young Investigator Award, even though he's over 40. But it's a really great pleasure to invite him back as an alumni of the CBR. I mean, he's been away for about 7 years now, and he's been carving a really nice niche for himself at the University of Calgary, across at the start of the Trans Mountain Pipeline. So, Tony, or near the start of the pipeline. So, Tony, thank you for coming, and looking forward to your talk. And
Great, thank you. Everybody can hear me well with the microphones, good? It's really great to be back and see a lot of friends, and some people were not here when I started. I have a student, Maggie, who works with Kelly McNaghy, who was with me, and I convinced her to come to UBC because I had such a good time. So it's always, well, thanks Maggie for coming, but it's always good to be back, see a lot of old friends, see a lot of ongoing collaborators as well. And I remember it was just under 7 years ago, I had my last week in the lab. So I ran 12 Western blots for a paper rebuttal. And then I had to walk out by Chris's office on my way out. So that was, well, just under 7 years ago. And it's always kind of scary because you're going on your own and you're starting with no people and nothing in your lab. And Chris, I remember the last discussion we had is like, don't take chances, have fun, try to do creative stuff. So hopefully you'll see some of that today. We've been having a lot of fun in Calgary, trying to do something maybe a little bit different. And a lot of it has not worked, obviously, but a few things have worked. And that's what I want to share with you today. I'm going to tell you 3 different vignettes, 3 stories, and they may seem a bit disconnected.
It's pretty loud. There's a little button down there.
It's right beside.
It's on the device itself.
Okay, is that better this way? Okay, sorry. Okay, good. I want to tell you 3 different vignettes that may be disconnected, but really the goal of our team is in the next decade to really bridge those ones together so we could do single patient personalized medicine on the single cell level and look at PTMs in that level. So we're not there yet. But we came up with some sort of an introductory that we call TEMPO. TEMPO is an acronym. In about 10, 15 minutes, I'll tell you what it stands for, and I'll tell you a little bit of the story of how this came about in 2022. The reason I put an I, so I like Shiku Fukuda as an artist, Japanese artist that's passed away, but why I put an I never thought I'd be working in eye biology, but I've met a good friend of mine who's a retinal surgeon, and we've started finding cool things going on in the eye, and I think you can see a whole new world of science that is seen through the eye. At the bottom left, you can see our lab logo. So I'm really passionate about proteases. So you can see kind of an abstract Pac-Man there because you use Pac-Man to depict proteases. And in the A of the lab, that's a protease inhibitor or a little ghost that's walking around. And you know, when I left UBC, I had a bit of this vertigo. I didn't really know where to begin. And it's like, well, I really love games and I really like protease. So why don't I just create a human game of Pac-Man? So what we're trying to figure out is what are the rules of the game? And in this game, this human game of Pac-Man, the Pac-Man is the protease, so it's going around the circle. Anybody's played the vintage game of Pac-Man? I think people know what it is, right? Yeah. It's pretty brutal now, but it still kind of works what we're trying to do. So the Pac-Man is walking around and it's getting chased by the ghost. So those are the protease inhibitor. And the point of the game is to eat the dots or the substrates. So what are the numbers that we're dealing with in humans? So we have 473 human proteases that are active. There's 96 that are non-active homologs, but technically 473 active proteases. They're not found all the time in every tissue, but this is the maximum number we can find. We have 156 protease inhibitors. And how many substrates do we have? So interestingly, every protein may be victim of mitosis at some point in their lives. So basically every gene and proteins that we have in human is likely impacted by proteolysis. I'm not talking about microbiome, viruses, fungi, but those are massive numbers that are not included here. But we all know that the microbiome has a profound effect on our health and in disease. So that's a whole new game, and these numbers are huge. So, you know, being in Chris's lab, and so why do we care so much about proteases is that every protein will be victim of proteolysis. There's the second largest class of enzyme after the ligases. They correspond to about 3% of our genome. We still kind of have an understanding of about half of the protease in terms of what substrate they cleave. So that means that hopefully I'll have a job for a long time because there's a lot of things we don't know anything about them. And if you talk about bacterial virus proteases, then this number is way smaller. They're pretty good drug targets. Protease themselves can be drugged. Protease inhibitors can be drugged for virus infection, for cardiovascular disorder, for blood coagulation. So there's a lot of really solid drugs in there. So I do think that some of them, not all, but some of them are druggable. And then that kind of became the story of tempo, this whole gene to protein conundrum that I think some of us have been talking for years. So how many genes and proteins do we have? In our genome, we have about 20,359. And I say about because not every protein has been identified. There's still about 1,000, I think, that have never been identified before. We call that the dark proteome web, or, so this is quite interesting, but that's really not the point because all our proteins are decorated with chemical modification. So there's roughly 300 protease, post-translational modification, where protease is one of them, and phosphorylation, glycosylation, and so on and so forth. A lot of the drugs that we have are against these PTMs of proteins. So I think we still have a minuscule understanding of how the proteins are decorated and how they regulate biology. So that's definitely something we're interested to go after. That leads me to the second chapter, the Lisbon conjecture. Why Lisbon? Because this idea came about when I was walking with my friend in Portugal, in Lisbon. So before we met in Portugal for this kind of happened during COVID. So Jeff Bernatsky and Brian Yep in Calgary had this amazing data set of single-cell RNA sequencing of COVID patient and COVID negative patients that were on respirator with bacterial ARDS. So they had this really nice story, and then they were able to distinguish the different flavors of neutrophils and other immune cells, and those were significant transcripts that are different in patients. And then they have their paper in revision, and the reviewer says, we don't care, despite the fact that it's amazing work. We want to see the proteins. Are the proteins there? So they came to me. We ran 140 patients with COVID and bacterial ARDS, and that was the highly disappointing end to this. So we could only get these gene and proteins correlated, and they had found 15,000 transcripts in these patients. That's the only protein I could really give to them. So I was really disappointed and kind of annoyed with this results. It worked out, we were able to publish the paper, but I was really underperforming. And I was like, we need to address this. This is just so sad. And you're just missing all this data that you just can't see because you're dealing with plasma, which is a problem that most mass spectrometers have had for many years, right? And what are the reasons, what are the challenges, whether it's serum, plasma, CSF, is that about 20 proteins constitute more than 99% of the protein weight, despite the fact that we estimate that maybe we have about 8,000 proteins in our serum. The dynamic range is a massive problem. So a typical proteomics will identify between 400 to 1,000. This is in every single patient. We can get these numbers up to 2,000, but you're going to get a lot of missing value if you're dealing with human samples because there's huge heterogeneity. So let's call it 1,000. You have 1,000 protein, and people on the transcriptomics field, they'll get 12 to 18,000 gene transcript at a single level. So we're just not even close, and we just can't do this correlation. Now, you can say, well, I'll just use tissue then. It's like, sure, we've gotten really good at that. 3,000 to 6,000 in tissue. In cells, we can get in single injection 7,000 to 8,000 proteins now very easily, and you can get that to 10,000 proteins if you start playing around. But the problem with that, this is bulk proteomics. You don't know what protein is coming from what cell. And that could be very important if you're trying to understand what your drug is targeting or what cell is producing your gene or your transcript of your protein. So that leads me to June 27, 2022. So on the picture, you can see myself in Lisbon. This is Vinit Mahajan, I call him Vinu, and Jennifer Vu. So Jennifer Vu's a med student in UCLA. Vinu's a retinal surgeon at Stanford. So we're walking together in Lisbon. It's a beautiful day. What do scientists do when we're together? We bitch a little bit about papers and grants that we're not getting. And then we talk a little bit about the problems we can't solve. And I started telling Vinu exactly this, that, it's so disappointing. We get all these clinical samples and I just can't get the proteomes high enough to really get good biology. And what you get is the same stuff. You don't get the cytokines. You don't get the drug target. I like this picture because Vinu hates selfies. So Jennifer and I took way too many pictures, but we didn't know we actually come up with a good idea on that day. So that's really where everything kind of began in terms of the tempo thing. We didn't have a name yet, but it came a little bit later. So this was published in October. So it took us about 10 months to get this out. And on this picture at the bottom right, this is the picture we came. I really like art. So this is Georges Seurat pointillism. So what we want to illustrate is you can go with a very complex tissue or biopsy and try to understand on the single cell what is the different colors, what are the different flavors that's going on. We partner up with Alex, who's a good friend of us. So the three of us came up with 6 landmark projects to test this concept that I'll tell you about in a few minutes here. Vinu, being at Stanford, had a good amount of money, so he was able to kick things quite fast, which is kind of cool because we got this proof of concept that this kind of works. It's not great, but it kind of works, and we're able to get a little bit new ideas. So we need to pick some organs to test this concept of better understanding protein provenance on the gene and the transcripts. So we thought that the eye would be a good starting point because it's simpler than most organs. There's 50 different cell types. There's single cell RNA sequencing that's been well described for this organ. So we could use that to create an atlas to know what cells is coming with what transcripts. So on this picture, you can see Vinu and Julian Wolf, who's a fellow. So they were able to collect 120 aqueous humor and 120 vitreous humor from 120 different patients. And what we did, instead of using mass spectrometry-based proteomics, we use SomaLogic, which is an aptamer-based profiling approach. And you may have heard of Olink, which uses a double antibody kind of system. So the advantage of using that is that we're able to get 5,953 proteins quantified and profiled in 240 samples in 120 patients. Now the problem, there's a lot of limitation, is that you have an aptamer binding. So you don't have information about post-translational modification. We also don't know the affinity of your aptamer or your antibody. So that's the limitation. However, by using this approach, we're able to, we feel, get new biology that we've never experienced before looking at the eye and different disease. So what we're doing here, we're doing a proteomics profiling, and we're creating a tissue cell transcriptomic atlas based on what we know about the eye. So in this case, we had 57 different cell types, 82,000 cells. We can integrate these two and start doing data integration. So any new techniques, you need a cool name. If not, it doesn't matter, right? So we need to come up with a cool name. So I was visiting Vinu and his wife, Marianne, in Napa Valley. I really like wine. So actually where we found the name Tempo was right outside Opus One Winery. And this is outside their car. And that's where we coined the name Tempo. Tempo stands for the tracing expression of multiple proteins origins. So what that means is that by association, you can find what protein is there in your sample and what does it trace back to what cell can produce that. So how does it work? So how many genes and proteins can be traced back to a single cell origin? And the answer is about 1/3. So I'll give you an example. If I tell you what cells produce actin, you're probably going to say, well, most cells, if not all cells. But what if I tell you CD4, CD8? What are you thinking about? T cell. Neutrophil elastase. Neutrophils, right? What about CD20? Maybe a B cell. So we didn't really realize that about 1/3 can be traced back to only one type of cell that can do that. And this is in the eye. So now, is 1/3 going to be outside the eye? The answer is probably not. It's probably going to be more in the 25 to 28%. But 1/3 is just enough that you can get full proteome profile in a single cell level without having to go through a highly complicated single cell proteomics workflow that only yields about 1,500 proteins now. So this is a bit of a loophole. It's far from perfect, but you can still get pretty good resolution by using this. And this is what the data looks like here. So we can profile every 57 cell types in the eye and figure out exactly what unique transcript is produced. So if you get a protein, you know it has to be coming from this cell. You can validate that if you want to be sure as well, right? So this is for the eye, for example. So in our data, we found that 36 proteins are solely produced by rod cells, 44 only by horizontal cells, 56 only from cone cells. So by using that, you can basically have a unique single cell signature that you can look into patients. So obviously you wanted to know, well, this is all good, but can we actually get new information about different disease? So we decided to select different disease. So we wanted something that targets the rod photoreceptors, some about the retina and vascular process, some about the neurons in the retina. So the first disease we selected was retinitis pigmentosa. It's a rare genetic disease that we see a lot of cell death specific to the rods. What's crazy about the eye, I didn't know that at the time, but Vinu was telling me that they can do single cell injection for CRISPR delivery. So if you know what cell is driving a disease, they can actually go with a needle and CRISPR it out with a single injection. That was approved a few years ago. So there's a lot of potentials for human gene therapy if you understand what cells is causing the disease. So as a proof of concept, you can see on the top left, the rod cells are predominantly impacted. This is what we expected. It's a rod disease, so we knew this was coming. It also illustrates maybe the heterogeneity of the disease, that there's a lot of other cells that are also impacted by this death of rod cells. So in fact, about 39% of the specific proteome of rod cell is decreased in patient with retinitis pigmentosa. We can zoom in specifically on the cell type that you want, and you can see, okay, what is in green, those are a normal, healthy individual. In gray, those are retinitis pigmentosa. So what are the proteins impacted? So if you're working on a drug target, you may be able to know what cells express that protein, and what other potential proteins can be impacted by a specific target you're interested in. So this was published a few months ago. So there's a lot more information. I just want to highlight another example of what we showed here is Parkinson's disease. And another advantage of using this, obviously you cannot get brain biopsies from living patient. So there's no alternative if you want to sample a patient with Parkinson's. So looking at the eye is one way, is kind of the window to the brain, if you will. And in fact, Parkinson's disease patient will experience vision challenges. They might have dry eye, they might have double vision and eye movement problems. So there's definitely features of Parkinson's that is detectable in the eye. So the way we're thinking about that, you can sample the patient before, you can give the patient a drug, and then you can follow up after. And with collecting sample in the eye, it's pretty easy. It's local anesthetic, so you can do needle in, needle out, and you can collect a significant amount of fluid that you can use for diagnosis or for understanding what's going on. So this was our data for Parkinson's patients. So you can see on the left, there's the healthy controls, and on the right, those are Parkinson's disease patients. You can see proteins that are upregulated, proteins that are downregulated. So going through this process, you may identify novel drug targets. If you already know what you're targeting, like you can see on the right of this panel, let's say your target is NSF. You want to say, I have an inhibitor to inhibit NSF. At the same time, you can see that NSF is produced by ganglion cells, amacrine, bipolar, and so on. So this also tells you if you shoot a drug in a patient, you should be hitting those cells, or you can trace it to these cells. We like proteases. So when you see proteases, you get excited. HTRA2 is a mitochondrial protease. So this is kind of neat because HTRA2 is solely produced by islocytes in the eye. This is a tissue-resident macrophage of the eye. So let's say you're developing a new drug, a new protease inhibitor for HTRA2, you're expecting to find it in tissue-resident macrophages. And that kind of gives me in terms of proteomic clocks. People have done genetic clocks, genome clocks, and are the things, and in terms of clocks, I'm not talking circadian rhythm, I'm talking long-term. How are things changing over time? So chronological age is an important risk factor. If we're older, we have higher incidence of disease. That just makes sense. The tissue aging does not correlate to chronological aging. Patients that have certain disease, let's say liver fibrosis, they have premature aging of their liver. So we kind of ask the question, in terms of an organ-wide, do we also have specific cell type aging? So single types of cell aging within a tissue. Do those have certain long-term proteomic clocks that are changing? So in this case, we've integrated the data. We had 120 patients. We did an age trajectory from age 45 to 90. We've done some machine learning and AI to find different protein aging cluster. And this is just a snapshot of it. So on the top left, you can see the senescent secretomes, which is correlated to 53 proteins in our data. Those can be traced back to macrophages, liver, and endothelial cells. You're probably going to ask, why is there liver cells in the eye? That doesn't make sense. In fact, there are cells that look like hepatocytes in the eye. We don't know where they're coming from. They're traveling from the liver or acquiring this phenotype in the eye. We don't know. What's cool is that, well, what's interesting, I should say, is that uveal melanoma eye cancer metastasized specifically to the liver. So there's this intricate connection with the eye and the liver. I don't think we truly understand why that is. So for example, for senescence, we can do tempo. We can trace it right back to those are the cells experiencing senescence during aging. But if you look at mitochondrial aging, no specific cell type can be linked to that. So that means two things. Tempo is really not working for that, and we see that often. It's not perfect. We can only trace back about a third of the protein. And also, maybe multiple cell types are experiencing mitochondrial aging. Same thing with DNA repair and telosome. So this is what the normal aging of the eye looks like. So you see multiple cell types that are impacted, and those are the proteins on the left that corresponds to progressive aging. Now, the next question is, can we detect premature aging in specific disease? So if you look specifically at vascular aging, we found that the retinal endothelial cells, RPE, and corneal blood vessel were impacted only in proliferative diabetic retinopathy and uveitis, but not the other eye disease we looked at. This one was really interesting to us, immune aging. Immune aging seems to be almost predominantly driven by B cells in diabetic retinopathy, retinitis pigmentosa, and uveitis. In fact, there's a subset of B cells that are correlated to aging, and those were kind of driven or changed in our patients. So you have a really premature aging just on these immune cells, but not so much on the other cells that we looked at. In terms of retinal aging, no surprise, retinitis pigmentosa was impacted by retinal aging, but also diabetic retinopathy patients. So again, we feel that we can really start understanding the contribution in different diseases that are driven by certain cell types. And this is what we're doing now. So I paired up with Julian, and this is our current paper that's in revision. And where we want to get at is illustrated here in this pretty complex slide. But what we're trying to do, this is patient with uveal melanoma or cancer. So we can see PEP3, PEP2, PEP1 in 3 different colors. Those are different clinical phenotype based on the proteome. On the right of that, you can see the healthy controls. In the eye, in uveal melanoma, there's been 30 clinical trials without much guidance. It was kind of, it kind of works in this cancer, let's try in patients. So only the PML inhibitors have shown potentially to work. And what you can see, every lane is a different patient. So you can really visualize the heterogeneity of the disease. There's not many identical patterns in terms of cancer patient. So what we'd like to get to is that with a single biopsy that can be done in a living patient, we can give this information to drug companies or to clinician to be what drugs would fit better your patient. So in orange, you can see elevation of a protein. In dark colors, you can see a downregulation. So if you look at the bottom left of the graph for ABL1, RAF1, PDC1, some of the patients not only don't have it, but they are downregulating this protein, and healthy controls have elevation of that. So some patients are giving this drug where they don't even express the proteins. It just doesn't make much sense in terms of going in. So if we could better inform the drug companies so they can conduct targeted clinical trials to patients that actually have the target, maybe we could see better profiling of what drugs should be given to a patient. So that's kind of one of our way that we're going to do personalized medicine. A lot of those protein here, you're going to realize those are kinase or phosphorylated kind of pathway. So we have not looked at phosphoproteome of this, and that's really the next step. Can we get actual PTM information per patient by combining the expression of these proteins so as the phosphopeptides targets of what these drug targets? So if you know what targets you're going after, what drug, then you can selectively look for these phosphoproteomes. So that's ongoing right now, and we're having a lot of fun with the reviewers discussing these ideas, as you can imagine. Okay, so let's change gear a little bit. Let's protease, because I like proteases. So being in the overall lab, I'm sure most of you are familiar with the TAILS technique. This was published before I started in the lab. You can see Leonard Foster, Jay's, Chris over here. We've been really using this technique more and more in biopsy. So actually Maggie's here has written this review that we're really trying to not just use cells, but really go after biopsies. Like I said earlier, there's a dynamic range that's complicated with biopsies. I'll just skip the workflow of how TAILS is done, because unless there's questions at the end, but I'm happy to talk about the details just because I'm sure a lot of you have seen these talks before. So I want to present 2 different case studies of how we're able to use this in patients in biopsies. So for this paper, I paired up with Ejafe Agbani, which I think he came at the CBR to give a talk, and Menchu Poon. So this is a disease called the Montreal platelet syndrome, kindred. So it's a von Willebrand mutation. We had 2 patients. So Menchu Poon is a hematologist in Calgary, and he had a mother and daughter that were suffering from this disease. So we're able to get platelets and samples from these patients. So these two women have this disease. It's a von Willebrand mutation. It's an extremely rare disorder. So it's kind of a clinical case that we're able to evaluate. They have microthrombocytopenia, spontaneous platelet clumpings. They also, one of their main phenotype is they have GI bleeding. And they kind of can treat it with von Willebrand concentrated infusion. It often worked. There's a few instances that it didn't work, and they were kind of a little bit worried about what's going on. But hearing about this, I thought it'd be a very cool opportunity to apply interim omics and to dig in a little bit about the mechanism of what's going on in these patients. So they have their high molecular weight multimers of von Willebrand spontaneously bind to platelets and are clear from circulation. That leads to accelerated proteolytic degradation by ADAMTS13. So when there's a protease involved, I'm always all ears. So there's something going on with their platelets, and we get new mechanism or to study what's going on in these patients. So EJAFE was able to get some freshly isolated platelets from these patients, and they're kind of already basally activated at all time. They're a little bit larger and they exhibit platelet clumps as compared to healthy. So on top in blue, you can see the healthy platelets, and at the bottom, those are platelets from these two women. So you can see that in the von Willebrand disease patients, the platelets, their size are a little bit bigger, and they also have increase in thrombin and some of the staining we've done. Now, if you look for platelet aggregation, and if you put them on collagen, the von Willebrand patients' platelets have reduced aggregation. So what we wanted to do is to profile these platelets and look at the different N-termini and see how different proteolysis may be impacted in these patients, maybe find a new mechanism of what's going on. So in the von Willebrand disease patient, those were some of the substrates that were cleaved differently than what we see in normal platelets. We found changes in focal adhesion. This is one example by fibronectin, CRK-like protein, and the von Willebrand factor itself is cleaved differently, which is kind of interesting and kind of explain what's going on in the phenotype of these patients. If you're looking at the proteome, so those are the proteins that are elevated or that should be normal in platelets, and those proteins were downregulated in these patients. So you can see, interesting, there's a lot of actin. So when we're trying to validate that, actin beta is severely decreased, so we needed some loading control. So we used a Ponceau dye, and those were equal loading, but you can see reduction in actin beta. We found also CLIC1, which is a chloride channel that is diminished, and we thought that was one of the part of the phenotype that's going on. And the CLIC1 knockout mouse have also platelet issues, so we thought that was kind of an interesting biology. And we can actually use NFA and changes and monitor the chloride influx in these platelets. So in blue, you can see the normal healthy platelets. If you add a fluorescent dye and look for calcium, chloride rather, you can see an increase. In the von Willebrand mutation patient, there's nothing changing. They're already at saturating. They're already problematic. That was an interesting, I think, novel mechanism that we're able to use with the technique that was developed in the overall lab. And again, in terms of what's the link with tempo, it would be very interesting that we can, get a better understanding of where these proteins coming from when you have these kind of disease, so you can better understand if you're developing a new drug, what is your drug going to hit? What is your drug should hit in these patients? In the other case, now we're going to change gear. I want to share a story that was published last year in our lab, and that's about osteoarthritis. So I paired up with Roman Krawitz, which his lab is next door to mine at the McKagan Institute in Calgary. And then we have three excellent PhD students, Bangshu, Louise, and Afshin, that have led this project. So in osteoarthritis, it leads to pain. You often see loss of joint lubrication. There's cartilage destruction and uneven destruction. There's swelling, and there's also inflammation. Not in all, but in some patients. So one thing that we found that was quite interesting is we started thinking in terms of mast cell. And what's kind of interesting is that mast cell levels are very low in most individuals that are not having allergies or asthma. But they're elevated in rheumatoid arthritis and osteoarthritis. And that's kind of a bit of a conundrum because mast cells are fast-acting cells. For anybody that starts sneezing, if you're in allergic reaction, it happens quite fast. But osteoarthritis and rheumatoid arthritis are diseases that happen over many years. So I thought that was kind of an interesting question to be like, how can you have fast-acting cells really have profound effect over decades of a disease? This is the data here on the x-axis. You can see the synovitis, so the inflammation of the knee. On the y-axis, you can see the number of mast cells. So if you have a worst case of osteoarthritis, you're likely going to find more mast cells in the synovial fluid of these patients. So we're trying to figure out what's going on there. Upon degranulation, mast cells secrete several proteases. So tryptase is one of them, kinase is another one. So tryptase is a trypsin-like serine protease. We have 4 in humans. There's the alpha that is inactive, the beta is active. We have delta tryptase and we have a membrane gamma tryptase. Most of the procellular activity comes from the beta form of tryptase. Tryptase assemble into a tetramer. There's no endogenous inhibitor of tryptase in our bodies. That's kind of interesting. No known inhibitors of that, but it needs to form as a tetramer and bind heparin. So Genentech, Roche has developed some cool drugs. They actually push away heparin binding to the tetramer of tryptase, having this kind of elegant disruption of the tetramer that leads to inhibition. They went to phase 3 clinical trials for the treatment of severe asthma, but they were unable to continue. This is the paper that I'm describing. So this is, if you are interested to know more about the biology of this drug and tryptase, is led by Bob Lazarus and Henry Mona and Genentech. I really enjoy reading this study. So if you want to know more about mast cell and tryptase, this is really a fantastic paper to read. And in the lab, Roman Kravitz was working on this proteoglycan, this glycoprotein called proteoglycan 4 lubricin. So this is what creates lubrication in our eyes, in our joints. In all our joints, we have synovial fluids, and PRG4 lubricin regulates this lubrication. We also have a part of our hearts that needs lubrication. So PRG4 has a key role in the heart as well. And you have a C-terminal amopexin-like domain and N-termini somatomidin-like domain. And it's a glycosylated mucin-like domain where this is where the lubrication happens with this O-glycosylation in the middle of this protein. So we tested, when I started, we tested all the proteases that I had. The only thing I had was 15 proteases. So Roman came to me. It's like, any other protease can cleave PRG4. I was like, I don't know, let's just dump it in. We did in 2 cleave, MMP3 and tryptase beta. So that kind of gave birth to this random project that we never really thought of it together, but I thought it was kind of fun. So we're able to reproduce that very multiple times with different preps of tryptase and PRG4. Within 5 minutes, you can see here a cut bend. It's very streaky when you run gels with this because of the glycosylation of PRG4. So this is kind of the best way you can get it. It can get messy quickly running these glycoproteins on gel. But you can see here in a time course that PRG4 is cleaved by tryptase beta. If we change the concentration of protease and substrate, we can also see a cleavage of PRG4. If we use a serine protease inhibitor, we can block this cleavage from happening. Next, we wanted to know where are the cut sites of PRG4 done by tryptase. So Alain Doucet and Chris's lab developed a platform or a new technique called atoms. So atoms, in a way, it's a simplified version of the N-terminomic tails. You can just add protein in a tube and see where things have been cut. And this is really helpful if you're looking at degradation because you can label the free N-termini after processing and see where things are cut. And this is one example. If you add tryptase to PRG4, you see multiple cuts. This happens rapidly. And those were the label N-termini we're able to find. I'll get back to this data later on in the talk. Those are 2 proteins in a test tube. So does this happen in human ex vivo? So I'll get back to this data a little bit later on. We've done tails on that, and I'll show you the data later on. So PRG4 is a lubricating molecule. We want to know, okay, if it's cut by tryptase, does it gain or lose lubrication? So we can use this as a apparatus and measure friction between the 2 surfaces. So it's a rod. You can put protein at the bottom and measure friction. We added tryptase. Tryptase is not a lubricating molecule. So what you see on the y-axis is increase in friction. If you add proteoglycan 4, it's a lubricating molecule. There's no friction. So it's flat. Now, if you add PRG4 and tryptase together, you can see there's a gain of friction. So that means after processing by tryptase, PRG4 loses its biolubrication properties. If you use a serine protease inhibitor, you can partially rescue that. We wanted to know in rat and mouse model, is tryptase get near PRG4 in the joint? So there's multiple models that we've tried. This is one of it. It's a destabilization of the medial meniscus. If you stain for PRG4, this is the cartilage, this is the bottom of the knee. It's like you're taking a pen and you're drawing a line. It's like you're taking a marker. It sits right at the cartilage surface. In red here, we can see NF-kappa B. So this is an uninjured control. There's no inflammation. If we induce this model, which resemble part of the steps that we see in human OA, you can see ruffling of PRG4 being lost and degraded. And you can see in red, increase in NF-kappa B. Now, if you, at the same time, treat with recombinant PRG4, we can partially prevent that. Now, the problem with this approach is that it doesn't last for very long. It will work for about a week, but then after that, you're going to see data similar as what we see in the middle lane here. So our concept or idea was that can we give PRG4 in recombinant manner in tandem with the protease inhibitor to enhance the effect of this, this protein injected. We think we can do that in direct injection in the knee of patient with osteoarthritis. We think we can give it actually in contact lenses for therapeutic in the eye because dry eye have a loss of PRG4. So we think we could use that as well as a therapy for ocular disease. That patient will suffer from dry eye. So adding more PRG4 would increase their eye lubrication. So we look into the joints of these animals in the DMM model. In this case, we colored the mucin domain of PRG4 in blue. So you can see the beautiful lining right in the cartilage where PRG4 sits. In green, you can see the C-terminal of PRG4. So the antibody just can't reach. It's probably buried in the membrane, so we just can't get any staining. In red, we can see tryptase, which is not present here because this is an uninjured control. So if you create an injury and 1 week post-injury, you can see there's loss of this membrane. What you see adjacent to it, this is actually the bone marrow that's leaking and it's starting producing PRG4 so it can repair the injury. And now, after 1 week, we can see the C-terminus of PRG4. And where PRG4 is located, that's exactly where we found tryptase as well. So we do think that these proteins see each other in vivo and they come at similar times. Now, 2 weeks post-injury, post-DMM, you can see there's a lot of destruction of the joints. And then already tryptase is going down because in this model, it's kind of neat. The rats, in this case, and the mouse, they heal back. And within 3 weeks, there's very little PRH4 and tryptase level. And 4 weeks, you can start to see reconstruction. It's probably hard to see, but in blue at the bottom left here, you can see that PRG4 is starting to go back in the lining and start going back. And if you look 8 weeks later, you can see now a joint that looks normal. We asked the question, well, if we add PRG4, what part of the inflammation would be impacted? So what we found is that there's a modest but a significant protection against TNF-alpha and interleukin-6 in these animals. Like I said, the problem is that this lasts for about a week, but then if you continue or there's more injury, it comes back to where it was. So it's a temporary Band-Aid, but it's really not a solution. We knew that PRG4 binds TLR. So how does PRG4 impacts or increase inflammation? So we knew it binds TLR 2, 4, and 5. So we asked the question, if it's cleaved by tryptase beta, what happens to PRG4? Do we get more or less NF-kappa B activation? So we had HEX cells that were able to remove all TLRs. And what you see in blue here is a positive control of TNF-alpha, which triggers NF-kappa B. But PRG4 is unable to trigger NF-kappa B activation if there's no TLR. So it's absolutely TLR dependent. Now, if you only transfect TLR2 positive, you can see in powder blue the positive control TNF-alpha, and darker blue, you can see in the middle PRG4. And in purple, if it's cut by tryptase, you have a worse enhancement of NF-kappa B. So it triggers a worse inflammation if it's cut by tryptase beta. And in green is if we treat with the serine protease inhibitor, we're able to partially rescue this enhancement of NF-kappa B. So we saw that in TLR2, TLR4, and TLR5. So all three TLRs are signaling modules for PRG4. Now, pre-tempo, so that was about 6 months before we started working with tempo, we wanted to know, well, what are the cells that are producing PRG4? What are the cells that are producing TLRs? So we used data mining for 3 different single cell RNA sequencing data. So this were the cells in a joint injury model. And what we see at the bottom is that PRG4 is solely produced by synovial lining fibroblasts. This is what we knew. In terms of lining fibroblasts, they don't express TLR2 and 5, but they express TLR4. So in a cis manner, we think that the fibroblasts can signal and crosstalk via TLR4. If you're thinking in terms of macrophages, there's macrophages population A and B here that are defined by some gene signatures. So the macrophages express TLR2 in high level, so as TLR4, and to a lesser extent, TLR4. So depending on what's going on in the joints, it's either a lining fibroblast communication that works with PRG4, or maybe you have an interaction with the macrophage that would come. The next question we wanted to know is that, does this happen in humans? So osteoarthritis is a challenging disease because once the damage is there, it's usually often too late and you have a lot of joint destruction. So we wanted to mimic, can we do early OA? So we took healthy synovial fluids from cadavers, human cadavers, that have no sign of osteoarthritis. And we spike in recombinant tryptase beta in a complex environment, which is loaded with natural endogenous inhibitors and what's going on in the synovial fluids. And we asked the question, well, what does tryptase beta would be able to cleave in this context? So luckily we found PRG4. We found one cleavage site of PRG4 in the C terminus. And just coming back to the data I presented earlier, this is a cut site we found at lysine 1330. So we think this is a tryptase cut site, but in the test tube we found multiple others. So why is that happening? We don't have a precise answer. One thing we suspect is the glycosylation pattern is probably a little bit different from recombinant PRG4 than what you see in humans, especially with the heterogeneity. So you may have different glycosylation signatures that's happening in humans. Therefore, that could be a way that may prevent tryptase beta to cut in the mucin-like domain. Those are all the novel substrate of tryptase beta that we found. And those were linked to toll-like receptor, ECM, and immune system. So also interested to better understand these other substrates and what are their link in osteoarthritis. The next experiment we did was to compare healthy synovial fluids to patients that have advanced osteoarthritis. So at this stage, we're a little bit worried, is it too late? Will there be any PRG4 left? Because as OA progresses, you have loss of cartilage integrity and you have loss of lubrication, so there's less PRG4. So we asked the question, can we see PRG4 and is it cut if we can find it? So we did find it. What was puzzling to us is that the cleavage site that was found was different than in healthy synovial fluids. And again, we know that this could be cut by another protease. It's a possibility that tryptase can cleave there because we've detected this cut site using a biochemistry approach. And why is it different in advanced OA than in healthy? Again, this explanation of the different glycosylation of PRG4 is probably the most logical explanation at this time that we have. But the point I think we wanted to make is there is processing of PRG4 in early, in middle, and in late OA phase. So that's another thing too, that it could be in terms of therapeutic, if we can deliver fresh PRG4 that may be totally protected from this, from the protease that can cleave it, maybe we can enhance the quality of joint lubrication for patients with OA. So I'll just end on this. So the artist, so I got a bit of a long story of Roy Lichtenstein out of Oswego where I did my undergrad, so I won't talk about it, but I just wanted to show kind of the way we're thinking in terms of tempo and how we can do that to better address personalized medicine. So this is kind of a zoom-in view of a Lichtenstein painting. So you can see every dot here represents a cell, and we're trying to understand the big picture. What is these dots playing in the big scheme of a tissue, and how is this changing in disease? And the more information you get, you can zoom out and really get a different picture of what's going on in the tissue. And we can ask, well, we can visualize the heterogeneity from patient to patient and better understand what's going on due to disease. So that's kind of the vision of what we're trying to accomplish in our team in the next decade. So we'll need better single cell proteomics capacity, which is really happening right now we're in this era that we can get better data sets. So I think this is really going to help us to understand on a single level what a single cell expresses in terms of PTM. Maybe in 10 years, I'll be able to come back and tell you whether I failed or not, but that's kind of what we're thinking. It would be awesome if we could do that. On this, I'd like to thank our wonderful lab and our funding that allow us to have fun in the lab. And I'd be more than happy to answer any questions if you have any questions. Thank you.
That's great.
Yeah, great talk, Tony. I'm wondering if some of those deleted sites in CBR are conserved in mice.
Yeah, good question. It's about 80% identical. The cut sites I showed, I think they're conserved in mouse and human. We've treated human PRG4 because there's a company called Lubris in Connecticut that produces that. So we've used mouse model, rat model, and pig model for skin. If you use recombinant human, it helps and it prevents in all these animal models that we've tested so far. So it kind of, it works in cross species. Like I said, the problem is that it only works for a little bit of time. Then it stopped working. So that's why we're thinking maybe we can engineer either a new version of PRG4 that cannot be cleaved as easily, or just combination with a protease inhibitor to try to get more of a life of PRG4 in fluids. So that's what we're trying to do now. Yeah, in the protein, yeah. Yeah. Yeah, good to see you.
So a question to your results in terms of the up and down proteins, expression of drugs in aging. So this up and down, is it primarily expression level or actually change of degradation?
Expression level, because oh, okay. You mean it could be
change, decrease in degradation, lysosomal degradation down. So you get an agglomeration protein, which is
yeah. So we don't have evidence of that's kind of where we want to get at to do N-terminomics and profile these. So you're absolutely right. It could be storage degradation. What we're doing is bulk proteomics. So we're looking at kind of net expression of a protein. But if there's more degradation, if there's proteasomal degradation, if there's cleavage of something, you would be unable to see it just due to the platforms by Somologic or Olink. So it's baffling to me that because Olink was bought for $3 billion in October of 2023. And it's baffling to me that people have not understand that very point, that yes, it gives you a profile, but you have no information of how much PTMs or how much degradation you may have. And you'd have the transcript information, but whether you know whether the transcript is up and the protein is up is not 100% correlating all the time, right? So we don't know. And that's really where it would be great if you could use N-terminomics or other approach to answer these questions. Especially for proteases themselves, but also known substrate that we know they're modulator or cleave in inflammation. It would be nice to do that. But we can do that in living patient. You can do that in tissue very easily. But if you're looking at plasma, CSF, or vitreous humor, you just can't do that with normal proteomics. Not very well, at least. Hi.
It was wonderful to see you in the pod today. It didn't occur to me that you were giving a talk. I had no idea what it's great.
Thank you, Ed.
I was interested in everything that you spoke about today, but in particular the VWF disease Montreal. Am I incorrect that the list of proteins that you showed there were the binding proteins to VWF? I was unclear a little bit about what you were showing. So that was the VWF pulp.
Yeah.
Okay, so there was a whole bunch of proteins that were really interesting, but of course to me, C3 stood out. Now, do we know if that was C3b? Is that indicating a mechanism for which von Willebrand factor is getting cleared, and so is why it's
dropping really dramatically in Montreal disease?
So I don't know on top of my head, and this is published. So we do find some complement and termini that are different. So we can easily look it up to see which one are cleaved and where they are cleaved, and then you could see what C3 cleavage sites are there. So we could probably answer that from the data that's there. What I've shown in terms of focal adhesion is it's an enrichment term. Those were the proteins that are cut differently between normal platelets and the VWD Montreal syndrome. So it just shows that there's different proteolysis happening that probably correlates to the the effects we see on platelets. So I don't know on top of my head if the complement, but we can absolutely, all the table is there with all complements that are different and how differently they are cleaved in this patient. So we can look at them.
It's really interesting to treat to increase the von Willebrand factor in that particular form.
Yeah, no, absolutely. And again, I think it was biologically interesting. In terms of therapeutic, these patients will get von Willebrand like infusion, and usually it's enough to control their symptoms. So clinically, they're less interested, but I totally agree with you in terms of biology. It would be very cool if we could do that in these women. And yeah.
And in the principle tempo, you're assigning a protein to a single cell into the eye. How much protein leaks in or passages from serum into the eye through whatever barriers are replaced there? So if that changes things will
So we're doing tempo and glaucoma right now. The data is being submitted or it's ongoing. So we have about 200 patients with glaucoma. So that's ran by the team at Stanford, which is great. So I'm looking forward to do that. What we do see in the vitreous humor is there are cells there, but there's a lot of protein, and there's a lot of protein crosstalk. So in the case of uveal melanoma, we've seen a lot of traffic, if you will. So we detect cells that are cancer specific that are not present in the eye. There's 140 proteins that are only belong to uveal melanoma, and we can trace them in the eye. When you validate, these proteins talk to neuronal cells or retina, and they talk to other cells in the eye. So there's a lot more traffic than we thought. There are cells present in the humor, but not that many because it's kind of the serum of the eye. And there's a lot of proteins. Now, in terms of leaking and stuff, the eye is somehow kind of protected in some ways. The question is, what about when things are advanced disease and stuff, and then you have like keratination or fibrotic part of the eye and stuff, and then you may have even more exchange, but it's somehow protected. What we don't understand is this connection with the liver. So there must be in and out ways that either cells or proteins are navigating in the macro environment. And there's a lot of that. I don't think we really understand that. We've looked right now, we call them meso environments, which is not within the tumor microenvironment, but just adjacent. So in the extended tumor neighborhood that we're talking, and we found a lot of traffic happening right in the humor. So maybe I'm thinking also with the pressure of the eye and stuff, things can just kind of, things travel in the eye. And it's just, I think, a distance that's reasonable that things start communicating in terms of inter-cell communications.
Do you see much amyloid protein, either beta or protein?
So one thing I didn't show, we thought for the aging part in the proteomics clock, we thought Alzheimer is the way. So we looked at 30 patients with Alzheimer and we saw no sign of aging and no amyloid, which was striking to us. Why is that? We have no idea. So we saw a lot more traffic in the eye in terms of Parkinson's and Alzheimer's. So that maybe tells us that Alzheimer might be much more local than slightly more systemic. I have no explanation of why that is. So we did not find a lot, actually. And we thought in Alzheimer patient, those might be where you would see changes. We saw nothing. So yeah, I don't know why. Yeah.
Quick question: how do you obtain healthy control?
Yeah. So students, poor people. No. It's, sorry, it's not very. Yeah. There's people that would go for a normal eye exam, and there's absolutely nothing wrong with them, and then they can be healthy controls. Yeah. Yeah. And there's, yeah, well, also, because all these people like that I work with at Stanford, those are all in the clinic. So sometimes they practice, like fellows will practice on each other for local, so they can get healthy fluids this way too. I don't know if I'm supposed to say that or not, but yeah, so we have a biobank, because with all the surgeons there, there's not none of that, so we get a good diversity of patients. I think they got a biobanks of a few hundred samples that are healthy. So no sign of ocular or any kind of disease. So yeah, which is kind of a nice control to have, so we can profile what's going on in a healthy eye. Yeah.
It's actually amazing. You think of what you've shown in your eye with actually, but it's actually not too bad. I mean, it's the patients will have once a month injections for degeneration. What happened? Well,
You mean like going to the dentist overall?
And another thing is, if you have a one eye infection, they can take the other eye as control. But there's issues in that, because sometimes you may have both eye effect, but that's another way you can collect healthy samples. If you have just local issues with one eye, then the other eye is open for sampling.
Well, so it was great, and it's really good to see you and judged by some of the comments, a lot of other people. And what's really great is that we sort of admit that we all occasionally have some bad ideas.
Prooms, thank you.